I think we did all four. This one,
Yes, sir. Yes, sir.
XXY, XXX0, then ZWZZ. I told that this is the important, right? It
Yes,
has
sir.
been asked ZWZZ, right? And this one. One more thing is left. That is the female sex determination in that one. This one. Honeybee. That is important. That is also
Yes,
important.
sir.
Sex determination in honeybee. This one. Look. Actually, the male drone. They have just a single set of chromosomes. So, like human, honeybees are also diploid one, right? Okay, okay. Just wait for 2 minutes. Actually, I think the rest of it. Just wait for 2 minutes. Then we'll
Okay,
start.
sir.
Let's start. Right. So, what I was saying that the sex determination in honeybee is very important. So, the male are basically the male have only single set of the chromosome. So, like human, the honeybees are also diploid one. They are diploid organism. Means they have like we have the 23 set. In the same way, they have the set of 16. So, two set of 16. So female have two set of 16, and the male drone means that is a male, right? So male have only 16 chromosome, means male haploid. So in honeybee, females are diploid and males are haploid. So they form the egg. So formation of egg take place in queen. So every egg, we know that this is the it takes place by the process of meiosis. That is important. It takes place by the process of meiosis. But meiosis is not required during the gamete formation in male drone because they are already haploid, right? So they are produced by mitosis, right? So meiosis do not take place, and mitosis takes place, right? So that is the important thing you need to remember, that reduction division or mitosis do not take place during the male gamete formation in honeybee, right? Now there are two kind of the egg in queen. One of the egg which are not fertilized, they are unfertilized egg. From unfertilized egg directly develop into organism. This process in which any organism develop from the unfertilized egg is called parthenogenesis. So formation of drone or male drone takes place by the process of parthenogenesis. They are just haploid. That's how they are formed. They are fertile and they reaches to sexual maturity, right? That is their role. Now what happened? Again, the fertilization of egg take place. The next another egg get fertilized. Those are the fertilized egg. They make the diploid female, and female are of two type. Both of the female, there is a worker female and queen female, right? Both of the female are diploid, but there is a difference between the size of this one queen and the worker. Well, actually, worker are basically sterile. Their development stopped before sexual maturity. They do not reaches to the sexual maturity. That is also important fact that you need to note it down, right? Queen reaches to sexual maturity. Why it reaches to sexual maturity? Because there is a special kind of the honey that is called royal jelly. Royal jelly is eaten by this queen one, and the rest of the worker they eat the normal honey, right? Work the function of worker to make the hive and to bring the honey, right? And the function of queen to just for the reproduction, right? Male drone, they also defends the hive, and they also work for the honey collection, right? So that is the sex determination in honeybee. If you want, please take this screenshot. Clear?
Yes, sir.
Okay.
Yes, sir.
Now the next one, mutation. Look, very, very important. I said already yesterday that the today's class will be very, very important. Look, just let me off the camera because it's taking this space of the screen. Okay, so look, mutation. What does it mean by mutation? The change in the sequence of gene is called mutation. So every organism have two type of the cell in its body: somatic cell and germinal cell. Whole body cells of the organism, they are called somatic cells, right? The whole body cells are somatic cells. Germinal cells, only gametes are called germinal cells, right? Gamete like sperm and egg. So even in human body, only ova and sperm, they are the germinal cells, and rest of the body cells are somatic cells. One more thing. Mitosis always takes place in somatic cell, and meiosis takes place in germinal cell. Right. That is also important concept. We'll discuss it again in the chapter of reproduction, human reproduction part. Now, what happened? The mutation which takes place in normal cell, that is called somatic mutation. Somatic mutation are not inheritable. Means if mutation takes place at my finger, it is not going to inherit it in next generation like my children, right? Germinal mutation is heritable, right? If the mutation takes place in germinal cells, they also carry forward in next generation. Mutation is not a negative term. Mutation may be progressive, it may be retrogressive. Means mutation may help in evolution carry forward, which it may be advancement in evolution, and even in mutation it may be a retrogressive, like it may be backwarded as well, right? So mutation may be beneficial as well as harmful. Both of the things can take place. Now, so this was we talk about two kind of the mutation on the basis of cell, somatic cell and germinal cell, right? I want all of you to please note it down, like the way, or if you want to take this screenshot, this is your own choice. Whatever you want to do, you can do, right? Okay. Now, mutation can be of two types: chromosomal mutation and gene mutation. Previously, we used to use the term chromosomal mutation, but nowadays chromosomal mutation have been omitted. Chromosomal mutation is not a correct term because mutation takes place in gene, not chromosomes, right? So what we say this term? So chromosomal mutation, better it can be defined by a term chromosomal aberration, right? So we define it with as a chromosomal aberration. Chromosomal aberration can be of two types. Now here you need to focus. I want your 100% focus over here. Chromosomal aberration, chromosomal mutation. The better term for that, chromosomal aberration. Now we use the term chromosomal aberration. Chromosomal aberration can be of two types. First, interchromosomal mutation means interchromosomal aberration means the aberration between two chromosomes. Intrachromosomal aberration means where the aberration takes place within a chromosome, right? Now look, interchromosomal aberration, please try to understand. In interchromosomal aberration, here it may be of two types. Translocation have been asked previous year, right? And all these new aberration have been asked in multiple times, right? But recently it has been asked translocation, right? So what are the non-homologous chromosomes? Like we have 23 pair of chromosomes. So every chromosome is in pair. So if you talk, let's talk about the chromosome number 1. So chromosome number 1 have its own copy. So both are called homologous chromosome. But if say chromosome number 1 and chromosome number 3, they are not homologous. Why? Because the gene sequence do not matches. So translocation takes place in non-homologous chromosome, between non-homologous chromosomes. First, very important thing. In translocation, a part of chromosome cut down or from one chromosome and translocated to another chromosome. Look, Non-homologous chromosome sequence is different. 1, 2, 3, 4, 5. Here A, B, C, D sequence. Now this part broken down, right? It broke down from here and it is transferred to another chromosome. Look, now here the second part, gene number 2 is omitted, deleted. So deletion will take place in one chromosome. Now this gene number 2 deleted from one chromosome, it is added on another chromosome. The next chromosome, there is addition of one gene and the sequence is 2 A, B, C, D. So gene loss do not take place. Deletion takes place at one chromosome. So in translocation, it takes place between non-homologous chromosome. Deletion takes place at one chromosome and addition takes place in another chromosome. Clear? Is that clear?
Yes, sir.
Very good. Another one is duplication. Duplication, so translocation takes place in non-homologous, but duplication takes place in homologous chromosome. Look, A, B, C, D, A, B, C, D. The gene sequence is similar in both of. If the gene sequence is similar in both, it means they are the homologous chromosome, right? Homologous chromosome means same gene present and similar loci. A, B, C, D. A is deleted from this one and added in another one. So here it is deleted, but here it's duplication, duplicated. Two copies are present. So here sequence is B, C, D. Now here sequence is A, B, C, D, right? So duplication, that is called duplication. The most important thing, I said you the translocation itself have been asked in recent year. One more question have been, what is the CML? What is the cause behind the CML? CML stands for chronic malignant leukemia. Leukemia is basically blood cancer, right? CML is chronic malignant leukemia. It is a disease which is caused in human. CML is caused due to translocation in 22nd chromosome, right? Translocation takes place in 22nd chromosome of human. That's why CML takes place. This disease have already been asked in examination. So please take this screenshot of this one as well, right? Now we will call that, we'll study the next one, another one. Look, so this another one, this is called intrachromosome, right? Intrachromosome. When it takes place between two chromosomes, right? Here, sorry, intrachromosome takes place within a chromosome. What happened? Here you can see a deletion takes place. So in deletion, what happened? The gene X is deleted, right? X is deleted from here, right? So the sequence was XYZQ. Now there is only sequences YZQ. It means a part of gene, a part of chromosome lost, means gene loss takes place. If gene will be lost, the function of the gene will be switch off, right? So because of deletion takes place, this disease have also been asked many times. There is a disease called Cry-do-Chat syndrome. Cry-do-Chat syndrome takes place due to deletion in the 5th chromosome of human being. What is the Cry-do-Chat syndrome? As I told you, CML is leukemia, means blood cancer. What is the Cry-do-Chat syndrome? Any one of you can tell me, please? In Cry-do-Chat syndrome,
chromosome 5 or 6 something is missing, no? 5.
yeah, chromosome 5th, it is written over here. It takes place. What is the symptoms? Means how you can say that the particular child have this Cry-do-Chat syndrome?
They'll have like the really high voice, the children,
Right.
something.
Yeah.
Like,
actually, when yes, that's high voice. When a child cry, it seems a cat is crying. So actually, this cry do chat means it's a French term. Cry do chat means the crying of a cat, right? So when a baby cry, it seems that a cat is crying. So that is the symptoms of cry do chat syndrome. Normally, if deletion takes place, the human baby do not survive. This is the only example of deletion. Only example of deletion in human being. That's why it is very important, right? The only example of deletion where deletion in chromosome takes place and the baby survive. Otherwise, if deletion takes place in human chromosome, baby do not survive, offspring do not survive, they die. Now the next one is inversion. In inversion, gene loss do not take place. Look, A, B, C, D, E. The cutting takes place at two places. First at the B places and the lower part at the D places. Now it will rotate in 180° and it will rejoin. Now the sequence will be A, D, C, B, E. The only thing which takes place, that is the sequence of gene changes, but gene loss do not take place in inversion, right? Deletion and inversion both are the intrachromosomal aberration. Is it clear? So remember both of the disease, CML, and if you will go with the previous year question paper, you will get number of the question from this concept from this PPT only. Clear?
Yes, sir.
Okay.
Yes.
Iram, clear?
Yes, sir. Clear, sir.
Very good, very good. Renia, clear?
Yes, sir.
Excellent. Now, actual mutation is gene mutation. Actual mutation is gene mutation. You will get the question I've been asked from here as well. Now, there are two types of the nitrogenous base, purine and pyrimidine. Adenine and guanine are called purine, right? Pyrimidine, thymine, and cytosines are called pyrimidine. Even uracil is also, but that is a part of RNA. Here we do not require that. So purine is adenine, guanine, and pyrimidine is thymine and cytosine. And we know gene mutation are sudden; they take place sudden. They are stable, and they are inheritable. Chromosomal aberrations are not stable, and neither they are inheritable. Right? Normally, they remains confined and inheritable. They are mostly recessive, means gene mutation takes place, but it do not reflect in the organism. Right? Number of mutations, thousands of mutations are taking place, but they do not reflect. So they cannot be observed by microscope. Chromosomal aberration can be observed by microscope at the time of the cell division, but gene mutation cannot be observed by the this process, this microscope. They are of three types: substitution, deletion, and insertion. Look. Deletion and insertions are known as frame shift mutation. Why we call them frame shift mutation, I will tell you. Now come to the substitution. Substitution can be of two types: transition and translation. In transition, a purine, nitrogenous base, a purine is substituted by another purine, and a pyrimidine is substituted by another pyrimidine, like you can see here. Adenine, this in gene sequence is ATGGAC. Now here everywhere where is the A, the G will come, right? Now the sequence will be GT, GG, GT, GGG, Now the sequence will change, right? Here what happened? That purine is substituted by another purine, right? When we talk about this one, purine with the pyrimidine, now what will happen? Otherwise, the pyrimidine will be transferred by another pyrimidine. In transversion, a purine AA is there. Now this purine in place of the A, T will come, right? So everywhere where A is present, there will be T, right? So purine is substitution by pyrimidine. These mutations are called point mutation. Why? They just take place at one codon. They changes or just one codon, right? They are called point mutation. These are not, they do not bring any remarkable change in any organism where they take place, right? So these are called point mutation. In point mutation, the change only takes place only in one nucleotide, right? Remember, this have been asked, what is the point mutation? In point, if you want, please note it down. In point mutation, the changes takes place in point mutation. The changes takes place only in one codon or nitrogen bases. Now come to the deletion. In deletion, what happened? A nitrogen base get deleted. It changed the complete sequence of the DNA. Look, that's why they are called frame shift mutation. 1, 2, 3, 4, 5. Now what will happen? The T is thymine is deleted from here. Now the third place, guanine will come at second place. Look, 1, 2, 3, 4, 5. Now the guanine will come place at second place. adenine at third place. So the complete frame will shift and the entire codon will change, right? Because codons are the set of three, so entire reading will be changed, right? They bring out a large change. That's why it is called frame shift mutation. Complete sequence of gene change. That's why they are called frame shift mutation. The same way is insertion. In place of deletion, we have inserted a amino acid. Now, when you nitrogen base, when you inserted a nitrogen 1, 2, 3, 4, 5. When you insert, what will happen at the point of insertion? Here I have inserted. Now, what will happen? Every nitrogen base will shift right side, one place right side, right? So complete uh, complete frame will change. So these insertion and deletion are called frame shift mutation, right? They changes the entire gene sequence, entire frame, and they bring out the remarkable changes. But the substitution mutation, they are the point mutation, they do not bring any remarkable change. Tell me, is it clear or not?
Yes, sir.
Take this screenshot. Aberration and mutation both are very, very, very important. You have to remember all things, whatever written in this PPT. And when you revise it, open these PPT and note down at your own notebook, right? That will be the best. Another very important thing, pedigree. What is the pedigree? Pedigree analysis is like, suppose that my grandmother eyes were blue, right? From grandmother, this characteristic came to my father, but my father's eyes were not blue. This characteristic came to me from my father, but my eyes are not blue. But my daughter, she have a blue eyes. It means the gene traveled from one generation to another, right? The gene is traveling from a generation to another generation to another generation to another generation. We are reading out, we are tracking any genes, and we are making a chart of it, this family chart of this gene transfer. This is called pedigree analysis. Normally, we do the pedigree analysis for the diseases, right? Clear?
Yes,
So for pedigree analysis, you must have seen that we make some symbol, right? Symbol. So look, this square, empty square is for the male, which is not diseased, unaffected male. This dark square is for the male, which is affected with disease. Female, unaffected. This circle, this is female affected. Then this, if we make this kind of this one, it means so this one, this one, this one, this one, and it means unspecified sex. What is unspecified sex? What does it mean?
Transgender.
Transgender.
they'll have like both female and male traits, right?
Look, if you ask me, sir, how many kids you have? I say I have 3 kids. So there is unspecified sex because they may be 3 daughters, there may be 3 sons, there may be 2 sons, a daughter, 2 daughter, a son. So possibility, you cannot, I have not mentioned sex to you. It means unspecified. Clear?
Yes.
If you get the question that a man have like a parent have 4 child, out of them a girl is affected with the hemophilia, it means they have, the question have mentioned just the sex of one offspring. The rest of the 3 are unspecified sex. Then for one where it said that the girls is affected, you will make this circle, but for the rest of the 3 you will not make this circle, you will make this circle, right? This diagram. So that's how it takes place. Okay, this is just unaffected individual, affected individual. Here, this line, this horizontal line between this square and circle, it represents mating or marriage. If there are double horizontal line, it say that mating between relatives, right? Okay. If this is a cross, it means the death of organism will take place, right? This horizontal line represents the mating, and this vertical line, it represents the next generation, right? And here a child, a male child, which is affected with the disease. Here mating is done, and a female child which is affected with a disease. Here, a pair which is not unaffected have a male and a female child, both are unaffected. A male, a female, mating done. The next generation, a male baby and a female baby, both are diseased, right? So these are the symbols. Last but not the least, this symbol represents this one, this symbol. Sorry. This symbol represents when a circle and there is a point between here. This is called carrier female. Who are the carrier? Carrier are those who carry the gene of disease, but they are themselves not affected by that disease, right? Normally the carriers are female, only females are the carrier. One more important thing I want to tell you that 99.9% diseases which are the genetical or hereditary diseases, they comes from mother to their son. They are not transferred from the father. They comes from mother to son, right? And they affect the son, right? So 99% diseases, if mother may be affected or may be carrier, and they are transferred to, they make their daughter carrier, but they do not affect the daughter, but they make their son affected, right? So in males, 99%, 99.9% genetical disease comes from mother, not from the father. Take the screenshot of this one, then we'll do some practice. Taken?
Yes, sir.
Okay. Mendelian disease. The first disease is the hemophilia. What is hemophilia disease? Do you have idea what is hemophilia disease? Yes,
Blood
Renia?
clotting does not take place.
Very good. Blood clotting does not take place in the affected person. Right. Absolutely right. If the injury takes place, blood clotting will not take place in the affected person. That is called hemophilia. Right. It's a type of Mendelian disorder. It means it is transferred from a generation to another generation. Look, it is also known as the royal disease. Why we call it royal disease? Because it had been discovered in the royal family of England, right? Just a minute. The disease was discovered in the royal family of England, right? That's why it is known as a royal disease, right? There was a queen. You all must be aware with the name of this name. It's a very famous name. Queen Victoria, royal family of England. Queen Victoria have a son, and his name was Leopold. Leopold was the first person which was affected with this disease. Actually, no, there must be a number of the people affected with this disease, but this disease had been discovered in Leopold, right? The first time in the world, this disease discovered in the Leopold. Leopold was affected by this disease, right? It is also known as bleeder disease. No blood clotting takes place. One very, very important thing, which is the question for you, this one. It is recessive, right? It's a recessive, this recessive gene, right? This disease is a recessive gene, right? And it is a sex-linked disease, right? A female with this one, XH and XH, this will be the female which is diseased. If it is present in only one X, then this female will be carrier. Like humans have, males have, in humans, male have only one X chromosome. So if there is this XH and Y, it means the male will be diseased, right? Clear? I think the point should be clear. Why in hemophilia blood clotting do not take place? There is a factor which is required for blood clotting. Factor 8 or factor 9. factor 8 or factor 9, which is important for blood clotting, is not formed. Is not formed in these people. That's why blood clotting do not take place, right? So if you want the person, if the person will get injured, he or she will die, then what is required? We have to give the factor 8 injection of factor 8 immediately. Then the person can be saved. Otherwise, the every drop of the blood will like come out of the body, and the person cannot be saved, right? So next, now listen. A normal male, look, I have made a cross over here. Please try to understand. A normal male got married with a hemophilic female. Now what will happen? Now I have made the cross. What comes to the cross? First, X and XH. What will be? This will be the female which is carrier, right? This female will be carrier, XH and X, female carrier. Another, again I have the make cross with the another X, XHX. Another female is carrier. It means 100% girls will be carrier. Now I said the mother do not give disease to the their daughter. It makes their daughter carrier. It gives disease to the son. Look, XHY, the son, and it will be diseased. Another XHY, 100% female will be carrier. I have written over it. 100% son are diseased or hemophilic. 100% daughter are carrier. Right? Again, out of them, one daughter got married with a normal male. What will happen again in next generation, F2 generation? So this was the parental, this was F1, now F2 generation. What will happen in the F2 generation? Right? This was the F1 generation. Look, you will get these kind of the question. Now, normal, a carrier female or a female, a daughter got married with normal man. Daughter is carrier, again normal man. XXA, this will be the daughter, which is the normal. Now X multiplied with this X. Again, this will be carrier. Not again the daughter will be carrier. Now Y X normal son. Y XH, a son will be what? A son will be affected.
Disease.
So how we can say that 50% daughter are, you will get this kind of the question. Yes, Heather, any question?
Yes, sir. Can you please explain this cross again? I couldn't understand it.
From where? F1 or F2? From
Just
starting?
from starting, yes, sir.
Okay, no issue, no issue. I'll do it. Okay. Look. Should I make a fresh cross? I can do that. Look, let me make the same cross again for you. I'll make that. I said there is a question that there is a normal, there is a normal father, right? Father is normal, neither carrier nor disease. So for hemophilia, so what will be the gene? XY, right?
Yes, sir.
Now I said that mother is hemophilic. So what will be the hemophilic mother? What will be gene combination? For hemophilic mother, the gene combination will be XH and XH, right? That's how the mother will be hemophilic. Clear? Now let's make a cross. One is sperm, another is sperm. One egg, another egg, right? Now let's make a cross. Now if you will make the cross, you'll find. So first, what you will do? You will take this X and you will cross with this one. That's what we do, right?
Yes, sir.
Now what will happen? The combination will be X and XH. What is the outcome? This is a daughter which is what? Carrier. Clear?
Okay, sir.
This is carrier.
Yes, sir.
She do not have disease but carry the gene again. Now what we do, the cross in cross, I take this one and I will cross with this one. Outcome is what? X and XH. What is another daughter which is carrier? Right?
Yes, sir.
Now you will take this Y and you will cross with this one. XH and Y. What is the outcome? There is a son which is diseased. Yes? Again. XH and Y. What will happen? Another son is diseased. So what I said 100% daughter. There are 100% two daughter. Both of the daughter are carrier. Is that clear? 100% daughter carrier. Clear or not?
Sir, why is the Y sperm recessive over here?
Sorry, because
Why is
yes,
the-
this is the recessive gene. Recessive gene will reflect when there will be both capital T, small t. Remember? So recessive gene will represent itself when it will be present at the both of the X. Hemophilia gene is found only on the X chromosome.
Okay, sir.
Right? So recessive gene represent itself only in homozygous condition. Right? What is that? Here it's in heterozygous condition. In heterozygous condition, recessive gene will not reflect itself. Clear?
Okay, sir.
That's why this female is carrier. Clear? So 100% daughter carrier. Is that clear?
Yes, sir.
And what about the son? There are two son and both son are diseased. So I can say 100% son are diseased. Is that clear or not?
Yes, sir.
Clear?
So
Now-
can I ask a question?
Yeah, sure. Please.
Is it necessary to have 4 childs for the F1 progeny?
Is it necessary to have?
4 childs.
No, it's not. But whenever we make the cross, we make the cross for 4 child. It is not necessary. From here with the 4 child, we can expect the percentage. What should be the percentage? Practically, this is not possible. But for theoretically, for study, whenever we make cross, we make the cross for the 4 child, right? 4 of it, always.
All right.
Okay. Now, again, now what the question says? Question again says to you that one of the daughter, any one of you can take this one, you can take this one, both are carrier. One of the daughter got married with a normal male. Clear? I made this line. Now again, what will be the normal X, normal Y? Here, when I will make the gamete, normal egg and hemophilic egg, XH. Clear? Again, what will happen? You will take this X and you will cross with this one. Yes? So what is the gene combination? XX. This is the normal female, no disease in this female. Again, you will take this X and you will cross with this one. Again, there is
Carrier.
X and XH. Yes, carrier. This will be carrier. You will take this Y, cross with this one. XY, normal. Y, normal son. What about this one? X and Y, XH and Y.
Diseased.
This is very good. So now my question, what is the percentage of daughter which are affected? That kind of a question you will get.
50%.
0%. No daughter is diseased.
Oh yeah, it's carrier.
What is the percentage of carrier daughters?
50.
50%.
Very good. Very good. Okay. What is the percentage of diseased offspring? I remember, please. I repeat my question. What is the percentage of diseased offspring?
25%.
Very good. Excellent. That's what I want. Normally, so if they ask what is the percentage of diseased son, then you will say 50%. If they ask you the percent of diseased organism or diseased kids, so you will say 25%. This kind of the question, confusing question are being asked, and normally the student make mistake over here. You will get only this kind of question. Is that clear?
Yes, sir.
If you want, please take the screenshot of this one and practice. The same thing will be for what one? The same thing will be for this one, color blindness. Okay, same thing for the color blindness. I will tell you. First, go through the pedigree. Now I have represented the same thing in the pedigree. Look, a normal person affected with, married with a diseased female, 4 offspring, both daughter are carrier, both son are
Sir,
diseased.
for the carrier symbol, you showed that it would be a
Yeah,
circle.
you can, so you can do like this way, or you can say, you can do like this way as well. Both are the same thing, right? You can get the both of the symbol, right? Both are for the carrier. So look, one of the carrier daughter get married with normal man, right? One daughter will be normal, one daughter will be carrier, one son will be normal, and another son will be disease. That's how we represent in pedigree chart. Clear?
Yes, sir.
Yes, sir.
Okay. Iram, clear?
Yes, sir.
Very good. So now the same thing will be for the this one. Color blindness. Color blindness follow the same pattern. The only symbol will change. What will be the symbol? Symbol for that. Look, the symbol for that will be only XC. XC. It will be color blind female. XC. X. This will be what? Carrier female. XC. Y. This is the will be color blind male. It will follow the same pattern, right? What is the color blindness? What is the color blindness disease?
Sir,
And what kind of the
inability to
disease?
differentiate between red and green color.
Exactly, the
Affects
person
the
cannot
electric
differentiate between the red and green
concept.
color. They do not get their DL, driving license, right? And the same thing. This is also the recessive. It is a recessive gene, right? Disease gene is recessive, and this is also sex-linked disease. Same thing. Is it clear?
Yes, sir.
Yes, sir.
Okay, very good. Now come to the another concept, that is sickle cell anemia. Sickle cell anemia. So first come to what is the sickle cell anemia disease? Sickle cell anemia also a disease where what happened? It is not a sex-linked disease. First thing, it is autosomal disorder. It means the gene of disease is found on autosomal, not on X or Y chromosome, right? This is autosomal disorder. What happened with the sickle cell anemia? A person will have normal RBC at normal oxygen level. The shape of RBC will be oval, like this way, oval shape of the RBC. But at the deficiency of oxygen, RBC will change its shape, and their carrying capacity to the oxygen will reduce, and the person may die. They change the shape like this, right? So you cannot take these people at those places where oxygen is level is low, at upper Himalayan part. You cannot take these people, right? At the low oxygen level, they may die. This is called sickle cell anemia, right? Why? The change of the RBC at the deficiency of the oxygen, RBC changes its shape from oval to sickle cell shape, like this way. This is sickle shape, right? What happened? Due to change in the alteration of nucleotide, one nucleotide in which form the hemoglobin, right? The hemoglobin polypeptide forming nucleotide, the sequence changes. So what change the sequence? The change in the sequence, normal nucleotide, the normal nucleotide sequence is CTC. CTC code for gluten, right? In a diseased person, in place of thymine, adenine comes. So sequence become codon becomes CAC. In place of gluten, it code for the valine, right? So that's how this disease takes place, right? Remember the codon. CTC changes to CAC. So in place of gluten, formation of valine takes place. Only one amino acid changes in hemoglobin. In hemoglobin chain, only one amino acid changes, and due to that, RBC changes its shape. Is that clear or not?
Yes, sir.
Next disease is phenylketonuria, PKU. It is also a hereditary disease. It is also homozygous recessive. Same thing, homozygous recessive. It is also autosomal disease. One more thing, it is also a autosomal disease. What happened? In normal person like you and me, amino acid, the name of that amino acid is phenylaniline. Normally, phenylaniline changes, it's a harmful substance. Formation of phenylaniline takes place in our body. Now, this phenylaniline is converted into tyrosine. In presence of enzyme phenylaniline hydroxylase, this enzyme changes phenylaniline into tyrosine. But in a diseased person, the gene for coding this enzyme is not found. So what happened? This phenylaniline will not be converted into tyrosine. Hence, the accumulation of phenylaniline will take place. In a diseased person, for phenylketonuria, lack of phenylaniline hydroxylase takes place, due to which phenylaniline accumulate and this enzyme destroy the neurons of the brain, right? That is the PK. Is that clear?
So can you explain the meaning?
Yes. Look, in a normal person, what happened? Formation of enzyme takes place. That name is, the enzyme name is phenyl hydroxylase. But the diseased person, this enzyme is not formed. So what is the function of this enzyme? This enzyme convert phenylaniline into tyrosine, right? But if this enzyme is not present, then formation of tyrosine will not take place. What will happen? Phenylaniline will keep on accumulating in the body of that person. Due to accumulation of phenylaniline, this phenylaniline will reach to the brain, and it will start destroying the neurons, and the person, like brain, it will damage to the brain and nervous system, and person will die. Clear?
Yes, sir.
Okay. So all the disease were very, very important. One more disease, that is Down syndrome. Down syndrome, Klinefelter syndrome, and Turner syndrome. They all are important. Down syndrome generally takes place in these babies, newborn one. Down syndrome is also known as, it has been asked, Mongolian idiocy. It is called Mongolian idiocy or Mongolism. Why? Because the child look like Mongol people, right? Mongolian people. So what is that? There will be a small nose and mouth and ear. They will be smaller than normal size. Slightly bent fifth finger. This fifth finger will be slightly bent. Almond shaped eyes and skin fold covering inner corner. In inner corner, skin will be folded. Flat and round face, right? Short and stocky build, right? That is the symptoms of this syndrome, right? The person will have little intelligence, heart and other organ dysfunction. They do not normal function. Gonad and genitals are underdeveloped. Sex organs will be underdeveloped. It takes place due to extra chromosome in 21 chromosome number. So the total number of the chromosome will be 47. So it is an autosomal diseases. In 21st chromosome, there will be, normally we have The 21st chromosome should be like this one. There will be addition of one more chromosome at the 21st, right? So this is called trisomy, right? Means 3 chromosome at 21st chromosome. So this disease takes place due to trisomy at 21st chromosome. Tell me. Take the screenshot and tell me, is it clear or not?
Yes, sir.
Okay. Klinefelter syndrome. Look. Turner syndrome and Klinefelter syndrome. In Klinefelter syndrome, Turner syndrome, there is a XO, right? Means there will be, it is a sex-linked disease, right? So there will be full autosomes, all the autosomes, but in female there will be only one X chromosome. So one X chromosome will get deleted, right? So we say that 2n minus 1. So total number of the chromosome will be 45, not 46. Normally a female have 46 chromosome, 44 plus XX, but this female will have only 45 chromosome, means 44 plus X, right? There will be folded skin at neck, right? First important thing. Poor heart breast development, right? And rudimentary ovary development, no menstruation cycle, right? It will be sterile female, right? It will be sterile female. That is a Turner syndrome. Remember, this is 44 plus X. Total numbers of chromosome 45. In Klinefelter syndrome.
One autosomal cell is absent in her.
Sorry.
One autosomal cell is absent, right?
One sex chromosome is not present over there. All the autosomes are present.
Okay.
Look, 44 plus, it should be XX, but one X is omitted over here, right? Deleted. Clear?
Yes, sir.
In Klinefelter syndrome, a normal male have 44 plus XY, but there is a one more X, XXY. So there will be more female characteristic, right? Means trisomy at the sex chromosome, right? This is trisomy at the X chromosome, 44 plus XXY, means total 47 chromosome. The person will have normal intelligence, sterile male, right? A small testis and long leg, and it will have obesity, right? Breast development takes place in males. What we call this disease? Gynecomastia. That is called gynecomastia. So breast development takes place in a male, right? Poor beard growth, and from the front it will be baldness, right? So this is called Klinefelter and Turner syndrome. Please take the screenshot. Clear to all of you? All these 3 syndromes, they are very important. Whatever we have studied, it's very important. You will see the question from this. Clear?
Yes, sir.
Excellent. One more thing I want to say, pleiotrophy. What is pleiotrophy? Pleiotrophy have also been asked. When pleiotrophy and multiple allelism, there are 2 characteristics. In pleiotrophy, when a single character is represented by multiple genes, means multiple alleles. This is called multiple allelism, right? The single characteristic, that is a one blood group, right? So blood group is represented by how many alleles? These alleles, 1, 2, 3, 4, 6 alleles. So 6 alleles are representing the blood group. So there are 6 alleles and only one character. This is called multiple allelism. What is pleiotrophy? Opposite. When one gene represents multiple characteristics, that is called pleiotrophy. Single gene, more than one character. Clear? That's it. That you have to remember.
Your face color or hair color will be example for multiple allelism.
Which one? Hair color, skin
Yes.
color.
Skin
No,
color.
actually that is called quantitative inheritance. When sets of gene, that is quantitative inheritance. It's not a multiple allism. Actually, I forget, there was a gene which used to decide our number of the characteristic, like hormones decide and eye shape decide. So there are some genes which represent more than one characteristic. So that is called pleiotropic. Okay. Now, last but not the least, some previous year questions. In humans, sex is determined by, keep on telling me.
Yeah.
Sex chromosome of father. Very good. Excellent. So A will be the answer. Second, in codominance, I have told.
B. Is it B?
Exactly. Both gene represent heterotrophic way. Third number, question number third.
C. Ordnance.
In Clefty's model, C. Borderland. Very good. Very good. Excellent. How many persons? Now you can see that you can solve each and every question. Whatever we are studying, we are in the right track, right? What should be this one?
7.
7.
Non-disjunction. 7, right? Very good. 7. D will be the answer. A disease caused by autosomal primary non-disjunction is?
So what is non-disjunction?
Non-disjunction means during the telophase, the chromosome do not separate; they remain stuck with each other.
Klinefelter syndrome.
So the question number 5. So question number 5 answer is D. No. Not, it will not be D. Question number, a disease caused by autosomal primary non-disjunction. So Turner syndrome, it will not be disease. Sickle cell anemia. Just a minute. Let me check this one. What should be the answer of this one? Give me 1 second. I'm confused. What should be? Down syndrome is the answer. Let me recheck it. Yes, Down syndrome is the answer. So the answer will be B. And the 5th answer will be B.
Sir, can it be Klinefelter syndrome also? Because
Look,
also
in non-disjunction, what will happen? There will be a sperm will have one less and one more, right? And this is autosomal, means one more autosome, means total 45 autosomes should be there, right? Now come to the Down syndrome. What is in Down syndrome? 45 plus XY, right? One autosome is more, right? Okay.
Yes, sir.
Look, I have taught you the mechanism that causes a gene move from one linkage group to another called
Translocation.
crossing over. Crossing over. A linkage group means a part of chromosome goes to another. Crossing over. It is asking for the mechanism. So mechanism is crossing over through which I'll teach you crossing over again in the cell cycle part, right? A true breeding plant is B will be the answer. So that is already a trick. Look, incorrect statement with regard to hemophilia. Tell me. I have just
B.
C. This is no A. Sorry.
Right, A. It is not a dominant gene. Recessive, sex-linked recessive gene. A is the answer. The same cross, kind of the cross. Look,
But sir, in the hemophilia NCRT, it is written there is a cascade of proteins that do not.
cascade of, yes, cascade of protein.
So
Right.
that means there
Means
are a lot of proteins.
actually what happened in hemophilia, formation of two protein note, that is a factor 8 or factor 9. These proteins are not formed. It may be either factor 8 or factor 9. That is not formed.
But sir, then the answer can be B also, because it is written a single protein is involved in the clotting.
No. Actually, the hormones are made up of more than one protein, prosthetic group and all that, right? They are not. That is not a just single protein. So
Okay.
hormone, right? And you, as you can see, the we in the first line I told you that this is the sex-linked and recessive gene, and it is written it's a dominant gene, right? And you know, if in medical examination you will get this kind of the question where the both are correct, then you will get the benefit of doubt if you will attempt the question and if you will take the wrong answer as well. Then
Okay.
everyone who have attempted the question will be given now. That is called benefit of doubt.
Okay, sir.
But normally, generally, it do not happens. It may happen once in a blue moon. A normal vision man whose father was colorblind marries with a woman whose father was also colorblind. So if the father was colorblind, female, then female will be what? Carrier.
Carrier.
Okay. So that's how we have to think. The female will be carrier. They have their first child as a daughter. Their first child as a daughter. What are the chances that this child will be colorblind? What are the chances? 2 daughter,
0%.
1 carrier, 1 neutral. 0%. Yes, A will be the answer. So you have to make this kind of the cross. Mutation can be induced, gamma radiation, right? So this is the glimpse of the question that kind of the question you will get in your medical examination. And if you'll cover the NCRT, if you'll cover whatever I have taught you, this is sufficient. You can crack the question. Question do not comes from the outer space, right? They come from the syllabus only. Just people make a hype. So is this chapter clear, or if you have any doubt, please ask me right now.
Sir, can you explain the meaning of autosomal disorder?
Which one?
Autosomal disorder.
Autosomal disorder. Yes. So first autosomal disorder is this Down syndrome, right? And what is the reason behind Down syndrome? There is a one extra autosome at the 21st number, right? So how many autosome? Normally a person have 44 autosome, but here 45, one more autosome is present, and that will cause Down syndrome. So this is an autosomal disease. Clear?
Yes, sir.
So at 21st number chromosome, there is a 3 chromosome. So I have written trisomy at 21st chromosome. Down syndrome, same in the is this one, but in Klippel syndrome, this is not the autosomal disease. Why? Because
The 6
yes,
chromosome is absent.
it's not autosomal disorder, right? And sickle cell anemia, this is also autosomal disease. Should I tell you? Is it clear? Phenylketonuria, PKU, autosomal disease. And this one, sickle cell anemia, both are clear. Both are autosomal disease.
Yes.
Any more question?
Sir, when you were explaining the hemophilia's crossings, at that time, the XXH combination was a carrier female, whereas the XHY combination was a diseased male. So why is why is there a difference between them?
Actually, you know, male have only one X chromosome,
Hmm.
right? So that's why in heterozygous condition, the gene will represent itself. Right?
Is
Another
it
X chromosome is not present. That's why.
okay? So is it that the X chromosome is dominant over Y chromosome?
Actually, X chromosome is very large chromosome than Y. Y is the shortest chromosome. So you
Oh.
can say rather than like we cannot say use this term dominant and recessive, right?
Yeah.
Otherwise, XY should be the female as well, right? But yes, X is very large chromosome. It have more number of the genes. Y is the shortest chromosome. It have the least number of the gene, right? So this is the reason behind it. Another X chromosome is not there. So that's why the recessive gene is this is the recessive gene, but in males at the heterozygous condition it represents itself.
Okay, sir.
Clear? Hiram, Renia,
Yes, sir.
any doubt?
No doubt.
No, sir.
Fine. So after the class, you will revise this chapter through the NCRT, and you will let me know if you have any doubt. Right now, this is the time to start the next. That's the molecular basis of genetics. It is the easier one. Molecular basis of genetics. This is the easier one. So first and foremost thing is that you will get the question. The question I have seen the question from this as well. Essential requirement for the genetic material. So on what basis you can say that this is genetic material or this is not genetic material? What is the basis to be like RNA is genetic material or not? So if anything, RNA, DNA, protein or anything, if follow these principles, these criteria, then I will say this is genetic. So what is the criteria for a compound to be a genetic material or a biocompound to be a genetic material? It should present in every cell. So this it should be present at every cell. Second, the amount should be same in all cells. That is true for DNA. It shows diversity. True. It should be able to duplicate itself, means divide itself without help of any other thing. DNA is the only thing which can do it. It should be able to pass its copy to progeny. Yes, that's what DNA do. Next, develop inheritable changes. It develop changes, means gene mutation takes place, and it is inheritable as well. Able to store information. That's the DNA can do only. Express information in progeny. So if these characteristics are present, then only we can say that is the genetic material. DNA have all these property. That's why we call DNA is genetic material. But yes, in the case of one exception is virus. In the case of virus, retroviruses, RNA is also a genetic material. Okay.
Yes.
Now the structure of DNA. Tell me, do you know the structure of DNA or should I explain it? Heather,
Yes, sir.
structure of DNA clear or should I explain it?
Yes, sir. Can you give a quick revision of it?
Yeah, sure. Look, this is a single DNA, right? When I say single DNA, it means the length should be 34 angstrom or 3.4 nanometer. You have to remember both of the unit. It should be in your mind. Sometimes they ask this simple kind of a question and student get confused, right? So I have seen a question. The question was, there are 200 nucleotide in a DNA thread. What is the length of that thread? 200 nucleotide pair means we can divide by the 10. 20 DNA. 20 DNA means what should be the length? 34 angstrom into 20. That will be the length. That's how we have to calculate quickly. but people get confused. Either they are if you are confused, then you can tick the nanometer, because the answer will be, the option will be angstrom in the nanometer both, right? So that's what sometimes student get confused, and sometimes they do the silly mistakes on these easier kind of the question, right? You have not to do that, right? So be precise. 34, the length of one DNA should be 34 angstrom or 3.4 nanometer. Width should be 20 angstrom, right? The DNA is the outer part of the DNA ladder-like structure. It is made up of sugar phosphate backbone. These are called strand. So every DNA have two strands. That's why we call it double-stranded. So I have said that this is double-stranded DNA, right? If one strand it is the 3 end, then there will be 5 end, and if there is a 5 end, then there is a 3 end. So both are opposite to each other. That's why we say that strands are anti-parallel. They are parallel but opposite. The strands are anti-parallel. Now what happens? The single DNA, it is twisted at 180-degree, right, and make two helix, right? It makes two helix. So that's why we say this is double helical. So DNA is double helical, double stranded, and anti-parallel. Now what is the chemical composition? These strands are made up of sugar phosphate backbone, right? So they are made up of sugar phosphate backbone. Sugar is, that is a pentose sugar, this one, and phosphate is S3PO4, right? This one phosphate, right? So this is the structure. One more thing, what is the charge on the DNA? Tell me, guys.
Negative.
Very good. Who gives the negative charge?
Nitrogen is base.
Phosphate. This one, look. Nitrogen and base are found inner side. This backbone is found outer side, and this H3PO4, this phosphate gives the negative charge. If you want, you can note it down. Phosphate gives negative charge to DNA. This question have been asked in AIMS. Again, the inner part is made up of, in one DNA, there are 10 nucleotide base pair. Base pair. So normally DNA have 4 kind of the base: adenine, thymine, guanine, and cytosine. Adenine also makes bond with thymine. Adenine always make bond with thymine, and guanine always make bond with cytosine. Adenine make double hydrogen bond with thymine, and guanine always make triple hydrogen bond with cytosine. ATGC, right? So there are 10 pair of nucleotide, right? So every DNA have 10 nucleotide base pair, right? And 34 angstrom length, 20 angstrom breadth, right? And this is the chemical nature of DNA. Is that clear?
Yes, sir.
Packaging of DNA, you can see. Look, first DNA get condensed. Condensation of DNA takes place. After condensation, the DNA wrap around a protein, right? So it make 2.5 circle of this protein molecule. This protein molecule is octamer. It is called histone. So DNA wrap around the histone protein, and this structure is known as nucleosome, right? When number of nucleosome, like this way, these are the nucleosome. These are the nucleosome. When number of nucleosomes are found in a string, this structure is called, this beaded structure is called chromatosomes, right? What we call this? Chromatosome. What is chromatosom? Question I've been asked. What is a chromatosom? Chromatosome is a string, a beaded structure that have number of nucleosomes. What is nucleosome? Nucleosome have a histone protein, that is an octamer made up of 8 molecule of histones, and a 2.5 circle of this one, DNA, right? Now this chromosome, it wrap like a solenoid, and this is called solenoid structure. This is it have a diameter of, or thickness of, 30 nanometer, right? Solenoid. What is the next structure? After solenoid, this solenoid compress, and the width becomes 700 nanometer, right? This 300 nanometer are compressed and folded into 250 nanometer wide, right? So every 250 nanometer, this is the width, under 250 nanometer. They wrap and make a 700 nanometer structure. This 700 nanometer structure is known as chromosome. So that is the final condensed structure. Now the important is the structure of nucleosome. That is very important. This is the structure of nucleosome. Nucleosome is made up of histone protein. So histone protein is an octamer, means have 8 molecules. H2A, H2B, H4 and H3. There are 2 set of each. H2A have 2 molecule, H2B have 2 molecule, H2A have these 2 molecule, H4 have these 2 molecule, H3 have these 2 molecule. So this is that's all called octamer. DNA wraps around it, right? And DNA, how the DNA wrap? DNA have negative charge, and histone is, what is the charge on histone?
Positively charged.
Very good, positively charged protein. That's why DNA wrap around. But this wrapping can slip down. So this wrapping do not slip down. So there is a glue. This is H1 protein, H1 histone protein. H1 histone protein, it keep this structure tightly packed, right? So H2A, H2B, H3, H4 and H1. 5 type of histone is found. You have to remember, not 4, 5 type of histones are present, right? Very, very important question. And normally students have missed this question. This question I have been asked. Histone is rich in lysine and arginine. These are 2 amino acids. You can note down if you want. Histone is rich in lysine and arginine. 2 amino acids. This have been asked in examination. Again, this is a nucleosome, and there is an extra DNA which linked with another nucleosome. This is called linker DNA. The linker DNA is the DNA fragment, DNA part, which link with another nucleosome. This length is of 200 nucleotide, right? So linker DNA and nucleosome having the 200 nucleotide long DNA. Clear?
Yes.
How many type of DNA is found? It is very important. The DNA which we are studying, what is that type of DNA? It is B type of DNA. So basically 5 type of DNA are found: A DNA, B DNA, C DNA, D DNA and Z DNA. The DNA which we study and most abundant DNA is B DNA. That's what we are studying. A DNA, it is right-handed, means its rotation is right-handed. It is right-handed. It have 11 nucleotide. But B DNA is also right-handed, 10 nucleotide. C DNA, right-handed, 9 nucleotide. D DNA, right-handed, 8 nucleotide. Z DNA is left-handed. This is the only DNA which is left-handed, opposite twisted, right? It have 12 nucleotide and its length is 45 angstrom. Take the screenshot. And jet DNA was discovered by Alexander Rich in 1979 in salivary gland of fruit fly Drosophila. I have written over here. Is that clear?
So what is that polytene chromosome?
Polytene chromosome, I'll tell you in cell biology. Actually, they are the very largest kind of the chromosome. They are largest size of the chromosome. They are found in the salivary gland of Drosophila, right?
Okay.
And they have puff at certain distance. I'll explain this polytene chromosome in cell biology.
Okay.
So you have to remember they are found in the salivary gland of Drosophila. So Alexander Rich discovered here, right? It's salivary gland. Next class, I'll start with transforming. We have to start from here, right? Put the remark that we have to start from the transforming. Okay. In case if you have any question, please ask me. If any doubt, please. The next class, we have to start with the Fine. So any question and doubt? No doubt? No question?
Okay.
Okay. So see you in next class. Till the time, study all this concept from your NCRT. Go through with your previous year question paper, and in case if you have doubt, you're most welcome to doubt. Okay.
Yeah. Thank you, sir.
Okay, Heather. Bye.
Thank you, sir.
Okay, Ram.
I wanted to ask, can we get access to this PPT of yours?
Access to this PPT.